Hi everyone. In this video, we're going to look at some more detail of the process of translation. If you haven't watched them yet, I recommend you pause here and watch the videos on the overview of gene expression and transcription first. If you look up a dictionary definition of the word translation, you'll probably find something like this one from Merriam-Webster, which defines translation as the process of rendering from one language to another. In the case of biological translation, the language we're starting with is nucleotides in the molecule of mRNA synthesized during transcription, and the language we're translating to is that of polypeptides, which are made of amino acids. Let's have a look at translation in action. It starts with the binding of the small ribosomal subunit to the mRNA and scanning to find the first AUG, which is the start codon for translation. Once that has been located, the relevant tRNA combines through complementary base pairing of its anticodon with the mRNA codon. And now, the large ribosomal subunit can be brought in. Inside of the ribosome, there are three distinct sites. The A, or aminoacyl site, is where the tRNA carrying the next amino acid will come in. The P, or polypeptide site, is where the polypeptide will be assembled and leave through a tunnel out of the top of the ribosome. And the E, or exit site, is where the empty tRNA will leave from, ready to be recharged and used again. We're now ready for the elongation step of translation. And we're looking at a simplified version here. There are other protein factors required for some parts, and the cell will also need to use ATP in addition to the components that we're showing here. Once the ribosome is positioned so that the tRNA is in the P site, the next can come into the A site. The amino acid attached to the tRNA in the P site will be cut off and attached to the incoming amino acid, and the ribosome will move, positioning the now growing polypeptide chain into the P site, moving the now empty tRNA into the E site. The empty tRNA can now leave while the next comes in, and the polypeptide chain is again moved onto the incoming amino acid. The ribosome again moves, the empty tRNA leaves, and the next comes in, and this process repeats, bringing in an amino acid for each codon until the stop codon is reached. Now the process changes slightly once the stop codon is reached, because of course we don't have a tRNA carrying a stop sign. Instead of a tRNA, the stop codon will be bound by a molecule called a release factor. This will use water to hydrolyze the bond between the final tRNA and the polypeptide chain, releasing the chain through the tunnel at the top of the ribosome and causing the whole assembly to fall apart. The polypeptide is now in its primary structure and will automatically begin to fold into its higher level structures, ready for processing and transport to its final location. Thanks for watching, and if you haven't had a chance to check them out yet, please watch the other videos on gene expression and transcription.